Hello, hello, greetings. Here, Maleeha Ajmal. Am I audible?
Yes, yes. Am I audible?
Yeah, you are audible clearly. Thank you so much for joining us in today's second class, Introduction to Biological Databases. Really interesting for me as well. That is, I hope that will be more interactive and meaningful lecture. So, no doctor, we will start within 2 to 3 minutes. Okay. Nurpur Akhtar, am I audible?
Uh, sure, who's gonna host today?
Yeah, you will continue within 2 to 3 minutes. Okay.
Okay, and what about the uh, attendance link? I think that we should provide attendance link between the session. It would be okay.
Just send me the Google Form link. I will share it.
Okay, sure.
Dear, Nurpur Akhtar, you can start now, please. Thank you.
Okay, thank you for giving me the stage. Assalamu alaikum and a very warm welcome to everyone. On behalf of Vetanov International Alliance for Science, I'm delighted to welcome all participants, distinguished guests, faculty members, and aspiring researchers to the Bioinformatics and Molecular Docking Online Internship Program 2026. We are truly excited to have participants joining us from different institutions and backgrounds, united by a common passion for science, research, and innovation. This internship has been carefully designed to provide both theoretical knowledge and practical insights into the rapidly growing field of bioinformatics and molecular docking. Before we begin, I would like to share an important announcement regarding attendance. A Google Form link will be shared in the live chat during this session. All participants are requested to fill out the form carefully to mark their attendance. As mentioned in the internship guideline, attendance is mandatory for certification, so please ensure that you submit the form before the session ends. Now, it is my great pleasure to welcome our speaker, Miss Maleeha Ajmal. She was the one who was our previous speaker as well. So, in her second lecture, we will going to explore the bioinformatics more deeply, discussing emerging opportunities as well as provide valuable guidance for making the most of the internship experience good for you. So without any further delay, I would like to call Miss Maleeha Ajmal again to take the floor and deliver her speech. Ma'am, the floor is yours.
Thank you so much, Noor, and good day, everyone, and welcome back. Am I audible?
Yes, you are audible and clear. You can start the presentation.
Okay, thank you. Okay, so yesterday we talked about what bioinformatics is. We talked about NGS, transcriptomics, and molecular docking. We talked about different tools and methods that make modern biology feasible. And I did mention some of the databases such as NCBI, UniProt, PDB, and TCGA. And today we will explore these databases in detail because here is something I want you, all of you, to understand from the very beginning of this lecture. The databases are not the background, they are not the supporting material. They are the foundation that every single bioinformatics analysis stands on. Before you run any blast search, the database has to exist. Before you drug into a protein, someone had to solve that protein structure and deposit it in a public repository. And even before you study cancer genomics, thousands of patients had to consent to having their tumor sequenced and that data to be made available. So, biological databases are the collective scientific memory of our field. They represent decades of experimental work carefully curated and made freely accessible to anyone in the world with an internet connection. A researcher anywhere in the world can access the same protein structure database. So, by the end of this lecture, you will know the major databases in bioinformatics, what they contain, how they are organized, and how to use them. You will also understand something deeper that data deposition and curation is also as important as the experiments itself. So, let's begin with what is a biological database? How do we define a biological database? So, can you move on the next slide? Okay, thank you. So, a biological database is actually an organized, structured collection of biological information that is typically stored in a computer system and it is made searchable and retrievable. That sounds simple, but there is enormous complexity underneath it. Think about what has to happen for a database to exist and to be useful. Number one, scientists have to generate data. They sequence genomes, they solve protein structures, they measure gene expression data, they run clinical trials. Then that data has to be submitted into a repository and that follows strict formatting requirements. And when the data has been submitted, then we have trained curators that are the biologists themselves. They annotate those entries, they add the functional information, they cross reference to other database, they add the quality scores, they add the taxonomic classification, and then the whole thing has to be updated continuously as the new data is coming rapidly and or understanding of existing entries is also improving. So Databases need to be updated continuously as well. Then a database needs a search interface, something that allows millions of researchers to query it efficiently. And the organizations that build the databases, the major one are the NCBI in the United States, we have European Bioinformatics Institute in the UK, we have DNA Data Bank of Japan, and they have been doing this for decades, and they're collaborating internationally, and they're making almost all of them freely available under open access principles. So now we will talk about some of the major types of databases. So if we speak broadly, biological databases fall into three major categories. First is the primary databases, then we have secondary databases, and then we have composite databases. So the primary databases, they actually contain original experimentally determined data that is directly submitted by the researchers. And some examples of such databases are GenBank, we have Protein Data Bank, we have Uniprot, we have SwissProt, and these are the examples of some primary database. And then we have secondary database. Secondary database are actually derived from the primary database through some computational analysis, pattern recognition, and curation. And the examples of secondary database includes Interpro, KEGD pathway, PFAM. These are some of the examples of secondary database. And thirdly, we have composite databases. They actually integrate data from multiple primary and secondary sources and form a unified resource. And examples of composite databases are Ensemble and String. So we will move through the major ones category by category sequence. Then we will talk about protein databases, then structure databases, then function databases, and disease databases. And I'll try to cover majority of the biological databases that are important in bioinformatics and from which all of you can benefit in the future when you're doing your research. So can we move on the next slide, please? Okay, thank you. The next one as well. Yeah. Okay, so we're going to start with nucleotide and sequence databases. So this is where we start because nucleotide sequences, we all know DNA and RNA, they are the most fundamental data type in modern biology. And before you can understand a gene, a genome, a pathogen, a cancer mutation, you actually need the sequence of that sample or whatever you are studying. So the number one database that we have in the nucleotide sequence database is GenBank, that is a part of NCBI, and NCBI is short for the National Center for Biotechnology Information, and it is one of the largest and most widely used biological data resource in the world, and it is a division of the US National Library of Medicine, and it maintains a family of databases that are used millions of times every day by researchers globally. And the centerpiece of NCBI is GenBank, that is the nucleotide sequence database. And GenBank contains hundreds of billions of nucleotide bases from millions of organisms. And every published sequence from any peer reviewed study must be deposited in GenBank before the paper is accepted. And that requirement is alone is why database has grew so explosively, because it is tied to publication pipeline, and if you have to publicize something, you have to first deposit that sequence into NCBI before you go for publication. So that is the reason GenBank is growing so rapidly. And GenBank is a part of International Nucleotide Sequence Database Collaboration, which is actually a tri-party partnership between NCBI, which is in the US, the European Nucleotide Archive (ENA) in the AML, EBI in the UK, and the DNA Data Bank of Japan (DDBJ). So this tri-party has a collaboration, and they all mirror each other. When you submit to any one of them, the data your sequence appears in all through. So this is the redundancy that ensures that no single point of failure can erase decades of scientific data. So whatever you store in ENA, that will appear in NCBI as well, and that will appear in DDPJ as well. Okay, so next we'll talk about RefSeq. So GenBank is comprehensive, but it is a bit messy because it contains everything that is submitted, including redundant entities. It also contains partial sequences and the sequences that have been revised since submission. So this is where RefSeq comes in. So RefSeq is short for Reference Sequence Database, and it is a curated, non-redundant collection of reference sequence for chromosomes, genes, transcripts, and proteins. And every RefSeq entry has been manually reviewed and annotated by NCPI staff. So RefSeq is also a part of NCPI, but it contains curated and reviewed information from GenBank. Okay. So whenever you want a reliable and authoritative sequence for a specific gene or genome, not just any deposited version, you can go for RefSeq. And we also have Ensemble, and Ensemble is actually a genome browser and annotation database that is also maintained by European Bioinformatics Institute (EBI), and where NCBI and GenBank are the primary sequence repository. Ensemble is basically a genome annotation platform. It takes the assembled genome sequences and it annotates them with gene locations, transcript variants, regulatory elements, comparative genomic data, and variations. So what it does, it takes a sequence and it marks where the gene is being starting, where is the transcript starting, where are the regulatory elements. So it basically annotates your genes and your sequences. So Ensemble is particularly strong for vertebrate and model organism genome. And if you're working on human, mouse, zebrafish, or any of the dozens of annotated organisms, you can use Ensemble, and it gives you a visual interactive way to browse the genome. You can zoom into a region, you can see all the annotated genes, you can click on a transcript, you can see its exon structure, you can compare it to the orthologs of that species. And for genome biology, this is a very, very essential tool. And Ensemble also provides Biomar, that is a data mining tool that allows you to export large tables of genomic data programmatically. So if you want a list of all human genomes, genes on chromosome 17 with their coordinates, geoterms, and uniprot IDs, Biomar can give you that in minutes. Okay. Then we have BLAST. You all might have heard the name of the BLAST. Can we move on to the next slide? Okay. So BLAST is like short for Basic Local Alignment Search Tool, and it is the most used tool in all of bioinformatics. That shows its value and importance of this tool. So what you do is you give it a sequence, any sequence you want to give it. It searches the database and it finds the sequences that look similar, and it tells you what is already known about them, and it ranks the hits by e-value, and lower the e-value, the more statistically significant the match is. And it has several variants: blast N for nucleotide to nucleotide, blast P for protein to protein, blast X for translating a nucleotide query to searching a protein database. And we have a full dedicated session on blast tomorrow. So today is just an overview, and I just wanted to you to know what blast is and what it does. So it actually takes a query. For example, if you have determined, or you have discovered a new sequence and you do not know what where it matches, you can simply put it in the blast search query, and it can match that sequence to all of the sequences that have been already discovered. And the blast is also incorporated in NCBI. So if you want to find any kind of sequence or gene on NCBI, you can find the blast option in the left corner. You can just click the blast, and It can give you the hits and exact matches between the query, and it searches the databases. Okay, so next we are going to move on the protein databases. Can we move on the next slide, please? Okay, thank you. So, as we know, the genes encode the sequences, but proteins do the actual biology. They catalyze the reaction, send the signals, they build the structures, they transport the molecules. So, understanding of proteins means understanding its sequence, its function, its structure, its interaction, and how it varies across the species as well. The protein databases are designed to capture all of this, right? So first we have UniProt, that is called the Universal Protein Resource, and it is the most comprehensive protein sequence and annotation database in the world. It is maintained by a consortium of European Bioinformatics Institute, the Swiss Institute of Bioinformatics, and the Protein Information Resource in the US. And it contains hundreds of millions of protein sequences, and it is the single most important protein database for most bioinformatics that are working in the world. And UniProt is actually divided into two sections, and the understanding the difference matters a great deal. The first section is UniProt KB, or we can call it SwissProt. That is the manually reviewed high quality section. Every entry in SwissProt has been reviewed by a trained biocurator who has read the primary literature, verified the functional annotation, checked the sequence, and cross-referenced it to the other databases. So SwissProt contains around 570,000 entries, and these are the gold standard protein records. And if you're looking for a reliable functional information about any of the well-studied proteins, SwissProt is where you go. The second section is UniProt KB Tremble. It is computationally annotated section. Tremble contains over 200 million entries, but their sequences are automatically imported from nucleotide databases, and they're annotated by computational pipelines. They are not manually reviewed. Tremble is enormous and comprehensive, but the quality is a bit lower. For a newly sequenced or poorly studied organism, you can use Tremble because it might be your only option, but you always have to check SwissProt if the function or the structure exists in SwissProt. Otherwise, you will have to use Tremble. And a UniProt entry is like a chest of information. For a well characterized protein, you will find the amino acid sequence, you will find the gene name, you will find the location, the function, the post translational modification. You will find a whole lot of things in UniProt. You will find the subcellular location, you will find the known variants, interactional partners, structural information, and it also has cross references to other databases, for example, geo databases and KEGG pathway, and all of this is for free in one place. Next, we have PFAM and Interpro. Can you move on to the next slide, please? Okay, thank you. So we all know that proteins are modular. A single protein is often made up of multiple functional units that are called domains. And domains are independently folding regions with specific functions. For example, the kinase domain activates the signaling. The DNA binding domain reads the DNA sequences. The transmembrane domain anchors the protein in a membrane. Understanding which domain a protein contains tells you a lot more about what it does. So, PFAM is a database of protein families defined by multiple sequence alignments and hidden Markov models. So, whenever you search a query sequence against PFAM, it tells you which known domains are present and where they are located in the sequence. Interpro integrates PFAM along with many other domain and family databases. We have Prince, ProSite, we have Smart, CDD, and others. They are all integrated into one database that is called Interpro. And an interpro search gives you a comprehensive view of all the functional annotations that different databases assign to your protein, and it is the most complete way to characterize a new protein's domains architecture. It tells you about the protein family, its domains, and many, many other things about the protein that you want to search. We also have string. In the last lecture, we discussed something about the hub proteins and how proteins are connected to each other. And we all know that the proteins do not work alone. They rarely work alone. They function in complexes, in pathways, in a network of interactions. So string is very short for search tool for the retriever of interacting genes or proteins. It is one of the most widely used protein-protein interaction databases in bioinformatics. String collects evidence for protein interactions from multiple sources. It takes the experimental data from co-immunoprecipitation and yeast to hybrid studies, and it also takes the computational predictions based on gene neighborhood and fusion events in other genomes, co-expression data, text mining of published data, and curated pathway databases. And each interaction is scored based on the quality and type of evidence that it has. So whenever you enter a protein of interest in string, you get a network diagram that shows that a protein and what proteins it interacts with, and it also have the thickness of each edge that represents the confidence in that interaction. You can expand the network, filter by evidence type, and you can also export that interaction data for further analysis. And string is particularly important for systems level analysis. The protein protein interaction network it provides is for uh, starting point for identifying hub proteins, running network-based drug target discovery, and understanding how a disease gene fits into a broader cellular context. Okay, then we have some of the structural databases. Should we move on to the next slide, please? Can we move on to the next slide? Thank you. Okay, thank you. Sorry. Okay, so knowing a protein sequence is one thing, but we also need to know the three-dimensional shape of the protein, and that shape is what determines how it interacts with other molecules, how drugs bind to it, and ultimately what it does in the cell. So we have protein data bank called PDB, which is a single global repository for experimentally determined three-dimensional structures of proteins, nucleic acids, and their complexes. And it was first established in 1971 with just 7 structures. And today it contains over 220,000 structures, and it is growing by thousands every year and every day. And the structures in PDB were determined by three major experimental methods. X-ray crystallography, which involves growing a crystal of the protein and measuring how it diffracts the X-rays. That was the dominant methods for decades and it still contributes the most to the structures. We also have cryo-electron microscopy, and it has become transformative over the past decade, and it allows researchers to solve structures of large complexes and membrane protein that resist structure and crystallization. We also have NMR spectroscopy that gives information about protein structure and dynamics in solution, particularly for smaller proteins. And each PDB entry has a 4-character accession code. For example, we have one HHO for hemoglobin. So if you want to search any of the entry, we can go to PDB and we can just search the accession number or the protein name. We can have the 3D structure of that, protein. And especially for drug design, PDB is indispensable. Before you can talk a drug into a protein, you do need protein's coordinates. You need to know more about the protein. And PDB is a database where you get those protein structures. Then we have AlphaFold databases. I did mention AlphaFold in the last lecture, but here I want to talk about it specifically as a database resource. In 2022, DeepMind and European Bioinformatics Institute released the AlphaFold protein structure database that was a collection of predicted protein structures for over 200 million proteins, covering virtually the entire UniProt database. And this is not the experimental data, these are the computational predictions. But for proteins whose structures have never been solved experimentally, which is the vast majority of known proteins, this is now the best structural information available. The accuracy of AlphaFold prediction for well-conserved protein cores is remarkably high, often within 2 angstroms of the experimentally determined structure, and the confidence is actually color-coded in the visualization. When you visualize the structure formed by AlphaFold, you'll find dark blue regions that will be of higher confidence. The yellow and the orange regions will be of low confidence, typically in disordered loops or terminal regions. And in practice, that means that if your protein of interest does not have an entry in the PDB, you check the AlphaFold database before concluding that the structural database is unavailable. For many of the proteins, you will find a high confidence predicted structure that is entirely suitable for molecular docking as well. Okay. So we also have some more databases for the proteins that are scope and cat. They are basically for structural classification. With hundreds of thousands of protein structures available, classifying them also becomes important. So there are two databases, scope and cat, that organize the protein structures into hierarchical classification based on their three-dimensional folds. Scope is short for structural classification of proteins, and it organizes the structure based on evolutionary relationship and structural similarity, and it groups them into families, superfamilies, folds, and classes. Whereas CAT, which is short for class, architecture, topology, and homology, it uses a similar hierarchical system, and both databases are useful for understanding structural relationship between proteins that might share little sequence similarity but adopt the same fold, that might suggest that a common ancestor or a convergent evolution led to those two proteins. So you may not use scope and cat directly very often, but understanding the structural classification exists, and it helps you to interpret the results when you find an uncharacterized protein that has a known fold, because the fold often suggests the function as well. Next, we move on some of the functional and pathway databases. Please, okay, thank you. Okay, so knowing the sequence or a structure tells you what a protein looks like, but what about the biology of that function? What does it do? When does it do? Where does it do? And in what context? And in partnership with other molecules. So functional and pathway databases answers these questions. So first database that we have is gene ontology database, and it is one of the most important conceptual framework in all of bioinformatics. It provides a controlled structural vocabulary for describing the functions of gene products, functions that are consistent across all species, all research groups, and all the databases. So basically, GO is an organized around three independent hierarchies called ontologies. First, we have molecular function that describes what a gene product does at the molecular level. For example, kinase activity, DNA binding, transporter activity. And the biological process defines the broader biological program that the gene product participates in. For example, the cell cycle regulation, inflammatory response, protein folding. And then we have the cellular component describes where the gene product is located. For example, nucleus, mitochondria, plasma membrane, and extracellular space. And each gene product in GO has a specific function that is associated with it. So basically,
Miss Maleeha, Mania, am I audible? Nurpokta, are you there?
I think she got network issues. We can take 1 minute to check if she's okay.
Everyone kindly stay in the live chat and also on the screen. She will join in 1 to 2 minutes.
You guys can fill the attendance form when until she joins, and after she joins we will resume our session again. Thank you everyone. Kindly be patient and stay with us. Miss, you were not audible. Kindly repeat again.
Nurpokta, did she join again?
I guess she got disconnected again. As we are waiting for our speaker, those who didn't fill the attendance form, they can fill the attendance form through the link. You can copy the text and then you can do it in your own browser as well if you are facing any problem. Um, Thank you for staying with us. Kindly be patient for a little moment as well.
No, we can continue and also please, Maleeha Ajmal, please inform after that presentation there will be a live class for a live demo, right?
Yeah, sure. Okay, I'm so sorry for the internet discrepancy. There was a power shutdown in my area, so I'm so sorry for that. And yes, we are going to have a practical demonstration where we will be studying the major databases, and I will show you how you can search for the relevant information using different databases. So we will do that after this theoretical part. So don't worry about that as well. Okay, so we were going with geodatabase, and I was telling you that if you want to search that a particular gene is involved in what kind of, what kind of a pathway it is involved in, what kind of biological process does it falls under, or the molecular function, or the the cellular component, we can go to geo and we can simply search the gene we want to study, and it is basically useful in gene set enrichment analysis tasks, which we do after dseq2 or edgeR. Okay, so the next database that we have is KEGG. The KEGG is short for the Kyoto Encyclopedia of Genes and Genomes, and it is a comprehensive database that integrates the genomic, chemical, and functional information to model the biology of cellular and organismal systems. And the most widely used component of KEGG is its pathway maps, manually drawn down diagrams of metabolic pathways, signaling pathways, and disease pathways, with each node in the diagram linked to the genes or compounds involved. And KEGG has pathway maps for hundreds of processes, for example, glycolysis, TCA cycle, MAPK signaling, apoptosis, and many, many more. So, whenever you are doing pathway analysis, which you will do extensively in RNA-seq and system biology work, KEGG is one of the primary database you will query. You will take your gene list and ask which KEGG pathways are enriched. If several genes in the same pathway, for example, the immune pathway, are differentially expressed in your tumor sample, that pathway becomes a mechanistic hypothesis for what is driving the cancer. And KEG also contains KEGG compound, that is a database of small molecules, and KEGG drug, that has information on improved drugs and their targets. For drug discovery work, the integration of molecular pathway and drug targets in one place, that is very useful. Okay, so next we have Reactome. So Reactome is another pathway database, but with a different philosophy from KEGG, where KEGG pathways provides a high-level overview. Reactome is actually a curated knowledge base of biological pathways at a mechanistic molecular by molecule level. Every reaction is a reactome is backed by a primary literature reference with specific proteins, small molecules, and stoichiochemistry, stoichiometry recorded. Reactome is particularly useful for human biology, and it is widely used in cancer research. For pathway enrichment analysis, run it against both KEGG and Reactome, and you can compare the results and you can see what kind of things are similar and what kinds of things are different in both of them. Then we have GO database, that is short for Gene Expression Omnibus. You might have heard the name of this database, and it is also maintained by NCBI, and it is actually a public repository of gene expression data. Researchers who publish their RNA-seq, microarray, or any expression studies are required to deposit their raw and processed data in Geo before publication. So you will find a lot of RNA-seq, microarray data if you want to do RNA-seq analysis or you want to do microarray analysis. Geo database is one of the best database where you can find the data for that one. And Geo currently contains data from over a million samples from thousands of studies spanning virtually every organism and experimental condition you can think of. Want gene expression data in Alzheimer's disease brain tissue. If you want COVID-19 patient blood data, if you want the drug resistant cancer cell lines, you can find everything in Geo database. And the practical workflow is you go to Geo, you search for your biological condition of interest, you can find the relevant data set, you can download the process data as well as the raw data. You can run your own differential expression in R, you can compare them to the other data sets, or you can use the data to validate the findings from your own experiments. And Geo is where you go when you need any real biological data to practice your analysis skills, and it is free as well. Next, we have some of other important databases that are disease and clinical databases. Can you move to the next slide, please? Next slide, please. Um, okay, I'll just start. So, the ultimate purpose of both biomedical bioinformatics is to understand the disease and to find out why it happens, how to diagnose it, and how to treat it. And that requires databases that link the molecular data to the clinical phenotypes. Okay, so the first database that we are going to discuss today is OMIM database, that is on online Mendelian inheritance in man. So, OMIM is one of the oldest and most respected sources in biomedical genetics, and it is a curated catalog of human genes and genetic disorders associated with them. Essentially, it's like an encyclopedia of the genetic basis of human diseases, and it is updated continuously by expert curators. And if you have identified a gene variant in a patient and you want to know whether it has been associated with a disease, OMIM is your first stop. An OMIM entry for a disease gives you the clinical description, the genetic basis, the chromosomal location, the molecular mechanisms were known, and the mode of inheritance, the autosomal dominant, recessive, X linked, and extensive references to the primary literature. So, for clinical genetics and rare disease search, OMIM is the foundational database. We have another database that is ClinVar, which is an NCBI database and that archives the interpretation of genetic variants and their relationship to the human health. And it is one of the central repository for clinical variant interpretation. When a clinical genetics laboratory sequences a patient genomes and it identifies a variant, for example, a single nucleotide chain, a small insertion or deletion, they classify it. Okay, for example, pathogenic, likely pathogenic, where variant of uncertain significance, likely benign, benign. So, ClinVar collects those interpretations from the clinical labs, research groups, and expert panels around the world, along with their evidence that are supporting them, and it presents it in form of one database. And this is important for several reasons. First of all, it aggregates interpretation from thousands of labs. So, if 10 labs have independently classified the same variant as pathogenic that is captured the uncertainty, many variants are classified as VUS and variant of uncertain significance because there is simply not enough data yet to show. So, ClinVar tracks how interpretation change over the time as evidence accumulates. So, for any work involving human genetic variation, GWAS, cancer genomics, rare diseases, ClinVar is a critical, reference. Then we have TCGA. Can we please move on to the next slide if you're there? Okay, thank you. So TCGA is one of the most comprehensive cancer genomics resource ever assembled, and it was a very large-scale multi-institutional project that collected tumor samples from 11,000 patients across 33 cancer types, and they performed multi-omics profiling of each of the sample. Multiomics, we discussed it yesterday a little bit, that we perform whole exome sequences, RNA sequencing, methylation profiling, copy number analysis, protein expression, clinical outcome data, all on the same tumor samples. And the breadth of TCG data is difficult to overstate. You can ask, what are the most commonly mutated gene in lung adenocarcinoma? Are there subtypes of breast cancer defined by transcriptomic profile? You can have any research question related to multi-omics or single omics using TCGA data without collecting a single new sample. You can answer those questions. TCG data is also freely available through the Cancer Genome Atlas portal or through the UCSC Xena, which provides a user friendly browser for exploring and downloading it. For anyone working in cancer bioinformatics, TCGA is a dataset you will come back to again and again. Then we have DB-SNP or DB-WAR. So we all know that human genomes differ from one another. Two people differ at approximately one in every thousand base pair. That is 3 million differences across the whole genomes. And many of these variants have no functional effect. Some change protein sequences, some alter gene regulation, and a small number cause disease as well. So DBSNP is the single nucleotide polymorphism database, and it is NCBI's repository for short genetic variants. For example, single nucleotide polymorphism, small insertions, small deletions. Each variant is assigned by an RS number that is a reference SNP ID. Whenever you see an RS in some number, the specific nucleotide change that cause a condition, and dbSNP contains everything known about it. And dbWAR is the companion database for large structural variants, copy number variations, inversions, and translocations, and variations that affect thousands to millions of base pairs rather than individual nucleotides. And both databases are essential references for any types of genomic variance analysis. Okay, so now I have discussed all the common databases or the major databases that we can use in any of the bioinformatics analysis. And after you have introduced to these databases, let me leave you with some of the few practical tips that will make you more effective from day one. But I guess we shall first do the practical session, and then we will go towards the some of the shortcuts or how you can, search the biological databases in an effective way. So let me share my screen with all of you. Okay, I'm not able to share my screen. Just wait a second.
Okay, take your time. Until then, participants will probably fill the attendance form. Everyone kindly fill the attendance form because it is mandatory for all. The attendance form is shared in this live chat, so kindly you can scroll through and you will get the attendance form and fill through this.
Okay, they are not allowing me to share the screen. I'm sharing my screen, but it is not
Oh,
able to just. Oh, okay, okay, wait a second. Yes, it's done. Okay, thank
okay.
you.
You're welcome.
Okay, so first of all, we will discuss the most commonly used database, that is NCBI, and we will particularly go towards the gen bank. And if you can see, this is the interface of ncbi. And here you have different databases that you can use within NCBI, but here we are going to use GenBank, that is a nucleotide database. So you can type any kind of protein name or anything you want to search, gene name. For example, I'll search BRCA1, which is a breast cancer gene, and I will just hit search. And you will have a lots of different variants of BRCA1 that will be Homo sapiens. As I already told you, GenBank has a lot of entries for the same gene or the same protein you are searching, because it's all the partial information, all the single nucleotide, everything that is published is deposited directly into NCBI before curation. So you can open any one of them just for the review. You can search your, if you are going towards research, you can search for RevSec for the most appropriate one. But you can, for now, we are opening any one of them, and you can see we have the different information about that gene. The organism is Homo sapiens, and the reference number, the authors that published this genome, and we have a lot of information about this here. And from here you can see that we can do blast analysis on the sequence right from NCBI. We do not need to go to another database. So that's how the databases are interconnected, and this is how they complement each other. We can do primer designings from the for this sequence from the same database, and we can pick the primers as well. So if you want, for example, the fastest sequence of this uh, gene that you want to use further on, you can just open this, the fastest sequence, and you will have it. Just give it a second. Okay, so this is the fastest sequence of the gene that we were trying to do. So you can simply copy it, or you can download it from here. Okay, so this is the NCBI GenBank. And if you see, when I searched here BRAC1, you saw this RefSeq. We have a separate database of RefSeq as well. We discussed that RefSeq is a curated database that has the most appropriate information. So if we want some kind of appropriate information, or we want to be sure that this is the right thing that we are searching for, we can go towards the RefSeq sequence. So we just open it, and it has numerous amount of information about the same gene, the organism name, the gene type, and the proteins it encodes, the transcript level, the summary of this gene. So it has countless information about even the different regions about it, and the rest of things as well. So this was the NCBI GenBank and the RefSeq database. Now we're going to move towards Ensemble database. So we discussed that Ensemble is a genome browser and annotation database. So we are going to search, okay, it's already searched. So EGFR is basically a epidermal growth factor receptor, and it is a major oncogene in lung and breast cancer. So we can scroll down and we can see that we have, this one is the EGFR complete gene, human gene. So we can open it. Okay, yeah. So this tells us that what this genes has, what kind of transcript levels it has, what is the biotype, is this a protein coding part, is this a nonsense mediated decay. So it tells more about the gene, what kind of locations it has. It tells us about the multiple transcript variants, different exon structures. So this genome browser tells you about the different, the splice variants and orthologs of the same gene. So you can explore that in your free time. For example, you can see what databases it also encodes. So you can even select one transcript ID and you can study it deeply as well. So you can do that in your free time. This is telling you from where it is starting, where it is ending, and what kind of it is representing. It's is it a coding part? Is it for example, this one is an exon, that is a coding part. Its length, its translational length is telling a lot about all the regions of the gene. If I do a little bit, summary about what ensemble does. Okay, I think I am clear. If you think you want to ask something, then we can do it at the end. Okay. Okay, then we have BLAST. We are going to study BLAST tomorrow in detail, but for now, I'm just going to tell you how you do BLAST search. For example, if you have a
Maleeha,
sequence, you just submit, you choose some, from here. You can choose different, you can, filter
is
different.
is my voice clear? Hello, am I audible?
Yes, yes, you're audible.
Sorry for interruption. Can you open that database from Google again?
Yeah, but it takes a lot of time. That's why I just opened them already. But yeah, I can open them again. Just a second. Okay, for example, if we are searching blast, we just search blast, and here we have it, the first database that we have. This is the portal of blast, and we can, for example, we have to search the human genome. We just type the human, and we can type any sequence here. For example, I am searching ATGGATGCA, for example. Like you will have the sequence you want to search, for example, a breast cancer sequence. So I will add it and I will do blast. So it is going to take a few minutes, 2 to 3 minutes, and then you will have this type of results, and it will tell you the sequence I entered. Because I entered a small sequence, so I will have multiple matches to that sequence. So the larger the sequence length, the fewer will be the hits or matches, and you can see where that particular sequence matches to. So blast is especially important when you have a sequence, you have done sequencing, and you do not know where that particular sequence matches. So you just put it on blast and you can get the name of the similar proteins or similar organisms where it was identified. This helps you to identify that particular sequence very easily. And here we have the e-value. E-value is basically the statistical significance, and the lower the e-value is, the more statistically significant it becomes. Okay. Okay, now let's move towards the protein databases. So we will first go with UniProt. I'm just gonna search UniProt here, and this is the database. And just let's put any of the protein. Here is an protein already listed. We're gonna go with the example P05067, and when we search it. Okay, so this is the amyloid beta precursor protein. You get the gene name, you get the status. Here you get the status, if it is Swissprot or if it's Trumbull. I already told you that UniProt has two parts. One is Swissprot, one is Trumbull. The Swissprot is the curated one that has been reviewed, whereas Trumbull is the computational one. So this one is the Swissprot. It means that it has already been curated and that is accurate. And then it also tells you the organism. So UniProt is very very extensive, and you get a lot of information about the protein from UniProt. You can know about the function of the protein, you can know about the features, what are the different binding sites of that protein, what are the different protein binding regions and different sites of that protein, how what they are doing, and you can even get their references if you want. And I already told you that UniProt is also connected with GO, so it also tells you what that protein is involved in different processes. different annotations, molecular functions, biological functions, cellular functions. So you even have those about a single protein in within UniProt. You do not need to go to Geo for, if it's an already determined protein, you can find that in UniProt as well. Then we have the enzyme and pathway databases. So we have a whole lot of information about the protein in the single database. So it is a very, very important database. And if you know what it tells you about, you can know a lot about your protein. For example, if you're doing docking, you should know about the protein you are doing your research in. So it is also about telling about its subcellular location. So it is telling a lot more information about a single protein, even the disease variants, what kind of diseases it is involved in that protein, and how it's involved in, with the publications if you want to, reference them. So it has a whole lot of information about a single protein, and it is not possible to cover that in one lecture. So today I'm just doing an overview of all the, databases. Okay. Next, we will also discover a very important database for proteins, that is string database. So I'm just gonna search string database, and let's search in any protein. For example, I'll type. You can search your, desired protein in the search bar. Okay, so strings gives you information like this. So you can see this was the protein we were trying to, search, and these interactions tells you which different proteins this protein is interacted with, because the proteins are all interacted. They do not function alone. They function in interaction with each other. So you can even pull this apart, and you can see how this is connected to all the other proteins, and how those other proteins are connected to all the other proteins. And if you select each one, you can even see here it has information about that protein. For example, this is the DNA repair protein and what role it plays. So you can even see which protein it is associated to, and what that protein is playing a part in. And this is a very, very important database when you are especially doing protein interactions. Okay. Okay. So now we are going to discover another very important database of proteins, that is AlphaFold, and this is basically the protein structure, determining a database. We are only going to discuss the major ones. I think we missed PDB. We will do that after this one. So you can search for any of the protein here. For example, I search grass, and here, okay, I have to select the specific one. Okay, so here you will have the different structures. Here is the predicted structure for this protein. What AlphaFold does, it takes the sequence from nucleotide database, and it predicts the structure of that protein. And we already studied that the dark blue color shows high confidence, and the yellow color shows low confidence. So you can, use it. You can fold it. You can see individual structure as well. For example, if you select this, you can get information about each part as well, how all parts are connected to each other. So this is the beauty of AlphaFold. You can determine the structure of any protein you want, and if you have the sequence written, you can determine the structure even if it is not available on PDB or UniProt. But of course, that would be your first choice whenever you are doing analysis. But if the structure is not available, you will always use AlphaFold for predicting the structure of that protein. So if we talk about PDB, we left that one, but this is the biggest repository to find the 3D structure of proteins. And for example, we will search any protein here. Okay, there is a protein. Okay, So this is the COVID-19 main protease complex, and this is the 3D structure of that protein. You can view it, we can explore it in 3D, and we can find structure, annotation, and everything about that. We can study about that protein, and it also has information about its individual units as well. So this is the 3D structure of any protein that has been discovered already. So you can get it through PDB. So whenever you are doing, for example, molecular docking, so whatever protein you have to dock it against, you will want the 3D structure of the protein with all the sections. So your first choice will always be PDB. If the 3D structure is not available here, then you will be going for AlphaFold to find the different to predict the structure of that protein if you have the sequence. Okay. Next, we will study some of the pathway. Let's study KEGG pathway database first. Okay, yeah, here is it. Okay, let's study any of the protein. For example, we have human signaling P53 pathway 04115. It's its accession number. Oh, sorry. Where did we go? Okay. Okay. This is taking some time. Okay, so this is the beauty of KEGG. It formulates the entire pathway of that gene. It tells you what kind of pathway that gene is involved in, what are the genes that are involved in that pathway. And for example, this is the P53 signaling pathway, that is the cancer repressing gene in humans. And a color coded pathway diagrams tells us that how deep P53 is activated by DNA damage, and how it is causing different kinds of hypoxia, nitroxia. It basically tells you about all the pathways this specific protein is involved in, and how different genes are also involved in that pathway. So this kind of analysis is very important when you are doing your differential gene expression, and after that you will have to see what kind of pathways your genes are involved in to have some kind of biological information about those genes. So now we are going to search for another database that is the biggest repository for the expression data, that is called the geo database. And from this you can import a lot of information about the RNA-seq data, the microarray data. You will just write any type. For example, I'm working on gastric cancer, I'll just write gastric cancer here. Okay, so I'm gonna go with the data ones. So this is the studies which have been done, and their data has been submitted on a geodatabase. You can simply click on any of the study that has been done on cancer, gastric cancer. You can study about that, how that was done, what was the design. This was done by CheapSeek, and you can also get the information, different information about that study, how it was done. And if you want to access the data, so here is the download option from where you can download the data and you can use it further for your analysis. For example, if you want to see what kind of samples have overexpressed genes and lower expressed gene, you can use this data to do differential gene expression, different types of analysis. So geodatabase has lots of information on which you can perform the differential gene expression, the NGS analysis, RNA-seq analysis, microarray analysis. So any kind of analysis you want to perform, geodatabase is one of the biggest repository where you can find the data. Okay, now we're going to move towards the disease and clinical database. And as we said, we have OMIM database first. Okay, so this is the OMIM database. and it actually tells you whether a specific gene has been identified associated with a specific kind of cancer or a disease. For example, if I have a gene BRCA1, I'm just gonna search it, and this is going to tell me all about BRCA1. How is it associated with diseases? We all know that BRCA1 is associated with breast cancer, but how is it involved? This tells us. You can, for example, BRCA1 is associated with DNA repair associated protein, so we can open it and we can study a whole lot of things about that gene and how it is involved in cancer, and how, what is the gene structure, what is the mapping. We can find different functions of that gene, so it has a lot of information about every gene related to the disease. For example, BRCA1 is a breast cancer gene. So for example, if you do differential gene expression, you will find different kinds of genes. So what you're going to do is you will simply put the name of the gene in this database, and you can find how that specific gene is involved in different types of diseases, and it is actually related to the literature that has already been identified. So it like kind of validates your research when you're doing your research and you want to want to see what kind of information your gene can provide about the disease. So this is the database. So another very important database that we have is TCGA database, that is the Cancer Genome Atlas program, and it has a lot of information about the cancer of the multi-omic data about the cancer. It has information about the data for about 33 types of cancer. So even if you want to do single omics, you want to study genomics, transcriptomics, you want to study any kind of epigenomics, you can go to TCGA. Or even if you want to do multi-omics analysis by integrating the data, you can go to TCGA. You can download the data. Here the data is accessible. Some of the data is not publicly available, but most of the data is publicly available. So you can access the data from here, and then you can here you can explore the data sets. It actually has three types of cancers. This is the cancer types that are being listed here. So you can get the data about all of these types of cancer from this database. So you can download the data, the kind of data, for example, if you want to download, but whichever kind of data you want to download. I'm not downloading it right now because it is a very heavy data. It is in GBs, so it takes a lot of space in your laptop. But you can access the data here from here, and then you can do different kinds of analysis. So these are the major kind of databases that I have covered right now, and I hope you all have understood about them. But if you guys have any query, you can even ask the uh, the instructor, or you can even ask here. I will answer them. And I highly suggest all of you to go and search these databases yourself and explore it yourself, because doing it yourself will give you more information about no matter how much I tell you about them. So I think it is better if you all go today and you search all these databases and you explore, and you'll find how amazing all the databases are. So once you're all introduced with these databases, okay, one thing I forgot, that whenever you're doing coding, for example, you're working on R or you are working on Python, you can access the data directly through the APIs. You do not then need to download the data manually. You can use specific codes and specific libraries that can download the data for you inside the Python or R, so you would not need to download the data yourself. And this is what separates a bioinformatician from a manual database user, because it is very hefty to download the data yourself. So if you have different APIs, you can download the data yourself. You just provide the link of the data, and that uh, you know the programming language can access the data through the API, and they can download it for you. So, at the end, we have covered a lot ground today. We have covered the nucleotide databases, protein databases, structural databases, functional and pathway databases, disease databases, and you have not just heard about these resources, you have watched them, and you can go and you can check these yourself as well. And I want you to do is that you can go and you can search for them. You do not just remember what you saw today. Go open NCPI, search a gene you know, your favorite gene, or one from the paper you have read. Follow the cross links, find this protein in UniProt, find its structure in PDB, find its interaction in string, and by the time you have traced one molecule through five database, you will understand this lecture far better than no amount of note taking could achieve. So that is all from my side, and thank you so much for today, for having me today, and I hope it was informative and you guys must have learned something. And if you guys have any questions right now, you may ask, and I'll be happy to answer some questions right now.
Thank you so much, Ms. Maleeha Ajmal, for such an insightful and inspiring as well as the practical hands-on training session. Now, as we are moving for the Q&A, I would request every participant to write your question in the chat box so that I can take it to our speaker. Okay, the first question we got, which is if we search for a mutated gene in the KEGG database, would we get the alternation in the pathway that's causing certain disorder? Miss Manihad, am I audible to you?
Hello, I could not hear you, Nooper. I'm sorry, my internet is, acting up. So can you please repeat what did you ask? I did not understand what you asked right now.
Okay, I am repeating it again, which is if we search for a mutated gene in the KEGG database, would we get the alternation in the pathway that's causing certain disorder?
Okay, so KEGG will show where the mutated gene sits in the pathway, but it will not automatically tell you how the mutation alters it or how it causes the gene, because it just tells you about that pathway. It will highlight your gene in all the pathways it is part of. For example, if you search BRCA1, it tells you all about the pathways it is involved in, but it does not automatically gives you does it because it does not know if your mutation is missense mutation, nonsense mutation, or deletion. So it would not tell you what kind of mutation it causes.
Okay. The another question is what is meant by that model confidence in AlphaFold protein database didn't get by that high blues or yellows?
Okay, so in AlphaFold, the confidence is the how much the database is confident about that structure, because AlphaFold is a deep learning based model. It is a computational model. So it tells you about the confidence level. How much is it confident that the structure it has given is correct? So the color indicates the confidence level. For example, dark blue color indicates that it is very much confident that this part of the predicted protein is correct. But the orange or the yellow part indicates that it is not very much confident what which kind of it is involved in, what if it is a correct structure or not. So this is what the confidence tells in AlphaFold.
Okay. The another question is it helpful for pharmaceutical sector or a student who is from pharmacy sector?
What are they asking about? What kind databases?
Now they're saying that it is going to help the pharmacy students if they learn about the bioinformatics and the alpha folds and databases.
Yes, of course. If you know, if they are going towards the data discovery, if they are going towards the drug designing or drug discovery, then bioinformatics is probably the only way, if they are going to deal with it. Because for drug designing and drug discovery, you need bioinformatics to do it quickly. So that's how it can relate to pharmaceuticals and pharmacists.
Okay. The another question is, you said that some type of data is not publicly available on database. What type of data it is and why it is not publicly available?
Okay, so there is a lot of data that is not publicly available. That might be some, kind of genetic data or the epigenomic data that the people do not, want to show. Some people respect the privacy and they just do not want to show that data in the databases because that are the open source databases and everyone can see those data. So that's why some of the data is not publicly available because of privacy reason. But we can, we can ask them if we want to access that. And most of the companies or the hospitals they allow you to access the data, but some do not because of the security reasons. And some people use the data for, the commercial purposes. So they do not allow to present the data for easily or freely accessible.
Yep, I hope that the participant got her answer. The another question we have that why does it make more sense to search Interpro before searching PFAM alone given what Interpro actually does?
Okay, so Interpro is basically an interconnected database and it integrates PFAM, ProSite, CADD, and several other databases. So it is genuinely very feasible to search first on Interpro because it gives us majority of information on the families, the domains of the proteins, and a whole lot of information that PFAM alone cannot give. So you just first search on Interpro and you can get the information about the protein families as well that the ProFam provides. So it is better that you just search on Interpro rather than going towards PFAM. And now PFAM is also integrated in Interpro. So the PFAM interface does not exist right now. We just have the Interpro interface where you can have a lots of information about a single protein.
Okay. There is another question is which database is more suitable for insects, protein function and interaction to other proteins?
Okay, for that we have string. String has a lot of information about protein-protein interactions and it contains millions of proteins. So if you want to study any kind of protein, any kind of organism, string is the way to go. And it is one of the best databases that you can use for protein-protein interaction.
Okay. The another question is that what is the main reason for blasting sequence?
Okay, so the main reason for blasting the sequences is that when sometimes you're doing wet lab validation or you might have discovered a sequence that has not been discovered before. For example, you are doing analysis on a rare kind of species and you might have discovered a sequence that has not been available or that has not been shown in NCBI before. So what you do is you just put the sequence in the blast and you search hit, and what you get is the matches to that data, to that sequence. So you can get the information about that sequence, a lot of information like which protein it encodes, what kind of different what kind of other species contain that sequence. So you can get a whole lot of information about that sequence. So if your, for example, sequence is already discovered, you can know about that as well. But if you don't, that would be a very novel thing that you would have discovered, and you can then publish your results, and you can give it to the NCBI that this sequence is not already been discovered, but I suppose this is involved in this kind of protein. So this is the
Mhm.
reason why we use blast.
Okay. Uh, we will take another last question, which is what is the lowest E value that shows the highest similarity?
Okay, so e value closest to zero is the best or the highest similarity, but we get the values because zero is almost impossible, especially it is possible only when we have a direct match. So we can get a value up to e to power minus 100 or e power minus 50. So these are the extremely significant matches in blast if the e value is extremely small, and if the value is greater than 1, that means that it is not that much significant.
Okay, thank you so much. And there's another question. It is the last question, and which is what are the criteria for choosing the best protein?
Okay, so that depends on your research question basically. So, for example, if you're doing your research on some type of cancer, for example, breast cancer, so you will have to identify which type of protein is the most, abundant or the most causing of the acquisition of that disease. So you cannot decide it yourself which protein is the most significant one, but you will have to read the literature about the disease you are studying. So this is how you know which kind of protein you are working on, but you'll have to do some research before that. You will have to study literature to know which kind of protein is best for you. And the best practice is you can do your own DGE analysis to see which genes are the most differentially expressed in the genes, and then you can use those proteins for drug discovery or drug preparations.
Okay, thank you so much again for answering the questions and giving the valuable insights. Your valuable knowledge, practical session, hands on training, and guidance have provided an excellent foundation to our participants as they begin this internship journey. We sincerely appreciate the time and effort you have dedicated to sharing your expertise with us today as well, like yesterday. To all participants, thank you for your active participation and enthusiasm. We look forward to seeing you our upcoming sessions of the Bioinformatics and Molecular Docking Internship Program 2026 as well. Have a wonderful night, stay motivated, and keep exploring the world of science and innovation. Thank you, everyone, and Allah Hafiz.
Hello, am I audible?
Yes, you are audible.
Okay, Nurpurkt, I have some uh, concerns related to the internship, uh, especially regarding the report, how to write the report. As mostly student asking again and again, like how we will write the report. As we organize the orientation, the purpose of orientation is also that like we are providing the guidance about the internship report, which is too much mandatory for your academic record. So, for example, you are taking that lecture tonight. You take the second class lecture. You must have take the screenshots of that lecture. Then what you will do, you will make a official professional post on your LinkedIn account, on your Facebook account, any account. Okay, after posting, must mention the organization. Our alliance members, our alliance council member will spot back you on your professional accounts, and after that you will prepare your report day by day. not at the end. You will prepare your report day by day on Google for on in the form of docs, and after that we have already shared a sample report, like how you will write the content that you learn from your lectures, and how you can take the data of our alliance, international alliance, from the given report, which is mandatory. Like you have to make the format as well as we shared the report in the group. So it is requested that everyone uh, Nurpakta, please take notice. There is one Asadali. I remove it from the group. And second is that after internship report preparing, uh, we will take that report at the end of internship journey, and on the basis of internship report we will issue your accredited international certifications. So thank you so much everybody for cooperation. And this is last reminder, those did not join the class, join the lecture due to valid reason, you have to contact after the joining class from recording lecture. We will provide the recording lecture, and after taking the class from recording lecture, just go to coordination with Miss Yusra, and she will provide uh, yeah, she will provide the attendance link, and in this way you can complete your attendance. So attendance and internship report, that are very mandatory component for your academic and this internship journey. Thank you so much everyone for joining us in this class. Best wishes from me, Ghulam Murtaza, and have a good day. Bye bye. Take care.